Hello, everyone. My name is Lydia Kariuki, and I'm a senior scientist and content marketing specialist at CloudLIMS. Today, I'm very much honored to be speaking at the Lab Roots Cancer Research and Oncology virtual event series. And in this session, we'll be tackling a topic that's very central and timely in the clinical research landscape, and that is the FDA Modernization Act 2.0. We'll be looking at how the changes introduced by this act are shaping clinical trials, as well as cancer research and oncology. And by the end of this session, I hope to have answered one question: what do clinical research labs need to do in order to adapt to these changes, remain competitive, and stay compliant along the way? So before we begin, let's first look at the core focus areas of our discussion for this session. We will first look at some background to give context to the FDA Modernization Act 2.0. If this is 2.0, we want to look at what 1.0 represented, what existed before this, and what are some of the major events that led to the updates in the current act. Central to this discussion are new approach methodologies. I'll be explaining what these are, what the implications are for cancer research, and why and how labs need to adapt. We'll also be sharing a tool at the end that labs can leverage in order to get ahead of the changes introduced by this new act. So let's begin with the background. How did we arrive at the Modernization Act 2.0? Journey began in 1938 when the Food, Drug and Cosmetic Act of 1938 was introduced. Prior to this, in 1937, there was the sulfanilamide tragedy, where a drug that had not been adequately tested for safety and toxicity was introduced to the market. This led to the deaths of about 100 people, and unfortunately, most of whom were children. There was a public outcry, which led the FDA to introduce this Act of 1938, which made animal testing in the preclinical phase mandatory. It's also important to note that this was not the first time that animal testing was being used in preclinical studies. If you remember quite well, in the 1880s, Louis Pasteur tested the rabies vaccine on dogs and rabbits. However, what this new act did was that it made animal testing mandatory. The Act of 1938 was modernized in 1997, and we had the FDA Modernization Act 1.0. Here, the requirement for animal testing was reaffirmed. However, between 1997 and the year 2022, some concerns had been raised about the use of animals in the preclinical phase. And this is what led us to the current Act 2.0. We'll be looking at these concerns in the next slide. But at this point, I'd like to mention that these are FDA Modernization Act 3.0 in the works, and this is supposed to advance non-animal methods in the preclinical phase. So let's look at some of the concerns that had been raised to do with animal testing. Animal rights activists had raised ethical concerns about the use, or as some would call it, the abuse of animals in studies that primarily benefit humans. In addition, there's been an issue of poor translatability of results from animal models to human models. It might surprise you to know that about 90% of drugs that make it in preclinical studies don't make it to phase 3 clinical trials. This is quite a high attrition rate, which brings us to the third concern, which is that animal models over time have proven to be time and cost inefficient. And the last concern is that with the emergence of new drug modalities, such as monoclonal antibodies, as well as advanced biologics, the complexities introduced by these new drug modalities are difficult to capture in animal testing. So these concerns are what led to the updates in the current Modernization Act, which is 2.0. So this act was enacted in the year 2022, and it updates the need for mandatory animal testing in the preclinical phase which were introduced by the Food, Drug, and Cosmetic Act of 1938 and later reaffirmed by the Modernization Act 1.0. While this new act acknowledges that animal testing has been foundational, new validated and animal alternatives are now recognized as alternative pathways for testing drug safety and toxicity. What are some of the major changes that this new act has introduced? But now you should be familiar with this. The need for mandatory animal testing is no longer there. This means that trial sponsors can now go ahead and seek FDA approval to conduct trials even without having conducted animal testing. Which brings us to the next point, what's the alternative? For the first time, the FDA now recognizes non-animal alternatives, and specifically new testing methodologies as alternatives for testing drug safety and effectiveness in the preclinical phase. And also, this update does not just affect small drug molecules, it also affects to biologics that are biosimilar and those that are biologically interchangeable. And how does the FDA intend to go about reducing animal testing in preclinical studies? In April 2025, the FDA laid out a roadmap to reduce animal testing. One thing is clear from this roadmap, the FDA now prioritizes the use of validated NAMs. In addition, this roadmap encourages collaboration across agencies such as the NIH. And lastly, it incorporates translational real-world human safety data from countries that have comparable regulatory standards. What this means in short is that where studies have been conducted on humans abroad, these results can now be used to inform FDA determinations. So this roadmap is a strategic stepwise approach to reduce animal use in preclinical safety studies. Now we get to the meat of our discussion today. What are new approach methodologies, aka NAMs? For starters, we know that these are non-animal alternatives. These are innovative methods that are designed to reduce, refine, or replace animal testing in the preclinical phase. Ideally, this method should offer faster, more human-relevant insights during safety assessment. It's important to note that not just any new methodology makes the cut. These methods must meet or exceed the scientific standards for animal testing. They must also prove to be effective in the scenarios for which they have been presented. In short, NAMs must be scientifically validated and fit for regulatory decision making. And with that background, we can now look at some practical applications of NAMs in the market today. And the first methodology we'll be looking at is organs on a chip. So what are these? These are microphysiological systems that use a technology known as microfluidic technology. So researchers or scientists take a device the size of about a USB stick, and this device, fueled by microfluidic technology, is lined with human cells. This device also has channels that mimic the flow of blood, as well as the exchange of oxygen and nutrients. And this gives researchers a realistic view that mimics the cell microenvironment, and with this they can be able to test out different drugs. Organs on a chip have some unique characteristics. One, they are 3D models. Two, they are able to mimic mechanical forces such as heartbeats or the rhythmic movements of lungs during breathing. And thirdly, they're also able to simulate chemical stimuli. So this provides researchers with a realistic, relevant model in which they can study both the pharmacodynamics as well as pharmacokinetics of drugs, of course, outside the patient's body. The second methodology we look at are organoids and tissue chips. Organoids are quite similar to organs on a chip. However, they do not require microfluidic technology. Here, researchers take cells and they provide them with a conducive environment that allow them to develop into miniaturized organs that have a 3D dimension. And this gives them an obvious advantage over two-dimensional cells on a petri dish. As much as these miniaturized organs do not encapsulate the full functionality of real organs, they're able to mimic the architecture and function of real organs. For example, an intestinal organoid will have villi-like structures and should be able to mimic peristalsis. The same breath, a liver organoid should be able to produce enzymes. So in short, organoids provide a scalable platform for studying drug safety as well as biology. The third methodology we look at are induced pluripotent stem cells. So here researchers begin with mature and well-differentiated cells. And through the addition of some genetic factors known as Yamanaka factors, named after the person who discovered them, these cells are reprogrammed back into a stem cell-like state. In short, researchers are able to create stem cells from mature cells. And this gives an obvious advantage over the use of embryonic stem cells, which come with ethical concerns. This model offers one significant benefit, which is flexibility, because researchers are able to re-engineer these cells to develop into virtually any type of cell. And because these cells are obtained from an individual, they carry the same genetic, molecular, biomarker profiles, making this a great tool for genetic disease modeling. In addition, these cells offer unlimited self-renewal capabilities. And this means that they provide researchers with a quote-unquote limitless supply of biological materials for use in the preclinical phase. So it's important to note that these same cells are used as the basis for organoids as well as organs on a chip. And the last method we look at is in silico modeling. Computer chips are actually made from silicon. So this informs the name in silico modeling. And here, computational methods that leverage AI and machine learning are used to create virtual patient cohorts in which researchers can carry out digital trials. This tool is very important because it allows researchers to pressure test thousands of ideas in a very short span of time. Like, for example, they can create a virtual cohort, and in this cohort, they can test out different drug modalities offered at different combinations, and at different dosages. And in this, they can study safety, efficacy, drug response rate, drug resistance. And this is something that would be almost impossible to conduct in a wet lab environment. So this makes this tool a very important one in supporting predictive toxicology. So at this point, one thing is very clear. Animal models have had their role to play in preclinical testing, especially as far as testing for toxicity and safety goes. However, as we move towards precision oncology, LIMS definitely offer a superior advantage. It will interest you to know that less than 15% of oncology trials progress beyond phase 1. And when you narrow down to murine models, the translation rate is less than even 8%. And this means two things. One is that it takes a very long time to carry out cancer research when we're using animal models. Patients in dire need of treatments often have to wait for a longer time, and some may die in the process of waiting. And secondly, this means the process becomes very expensive. And unfortunately, the cost is eventually transferred to the client, to the patient, making anti-cancer treatments relatively expensive. The third concern or limitation we have here is that personalized medicine exposes further limitations We're no longer looking at cancer as one general disease, but we're looking at it as a disease that is affected by individual factors such as one's genetic, molecular, biomarker, and even epigenetic factors. With this, it's very difficult to capture this complexity, beginning with an animal model. So with all this, I think the benefits of NAMs in cancer research should be quite obvious. For one, they improve predictability in oncology research, and we have seen this with in silico modeling, where researchers are able to pressure test thousands of ideas digitally. Two, this offers human relevant insights beyond animal models. And we have seen this captured very well through the use of organoids as well as organs on a chip. And lastly, NAMs support faster and safer therapy development. So let's look at some specific applications of NAMs in the cancer environment. And the first one we look at are organs on a chip. So here we said we are creating microphysiological systems using microfluidic technology. And in this case, we are using cells from the patient's tumor. So this model is able to replicate the patient's tumor structure and blood flow, providing researchers with a realistic model to study the tumor. For example, in the case of our breast cancer, the researchers should be able to look at how cells break away from the primary tumor in the breast, move through the blood or lymphatic system, and find their way to distant metastatic sites such as the brain or bones. In the case of a lung cancer, they should be able to study how breathing mechanisms affect the breaking away of tumor cells and even metastasis. So this creates a realistic model to study drug penetration, that is how drugs penetrate the tumor and how resistance mechanisms develop over time. If you're aware, you should be aware that drug resistance is one of the primary reasons why anti-cancer treatments fail. This model also allows researchers to study immune-tumor interactions. The second example, we look at our organoids for precision oncology. Here we have organoids where the cells are derived from the patient's tumor. And with this model, researchers can test multiple therapies outside the patient's body, that is ex vivo. And because the cells are derived from the patient's body, researchers are able to create treatments that are suitable and specific for this individual patient, making this a very great tool for creating personalized treatment options and supporting precision oncology. And the last example we look at are in silico cancer models. So again, here we are using AI and machine learning to create virtual patient cohorts, and researchers can run digital trials with this. For example, in the case of prostate cancer, researchers have been able to have created digital twins where the virtual cohort integrates information such as patients' PSL levels, tumor oxygenation factors, and imaging findings. And with this, they can test out different drug modalities. And this ensures that only the drug modalities that show the highest potential for efficacy and safety make it to the clinical trial phase. This makes this a great tool for predicting toxicity and efficacy profiles and also accelerating early phase decision making in a cancer and oncology landscape. So where do clinical research labs come in? So at this point, it should be clear that NAMs have the potential to reshape the cancer research landscape. Labs will have a pivotal role to play in making this shift from an animal first to a human relevant model realizable. NAMs will come with new testing methodologies, new instruments and specialized software that will need to be integrated, and of course, a diverse array of data sets. This will all end up at the lab level. And one thing is clear that for labs, operational changes are going to become inevitable. So in the next session, we'll be looking at what these changes mean at the lab level, and then what labs can do to get ahead of these changes. And perhaps one of the most significant challenge will be the sheer volume and complexity of the data that will be arriving at the lab level. For example, from organs on a chip, expect to have continuous sensor output. From organoids, imaging histological findings from induced pluripotent stem cells, those are genetic information, and from in silico models, expect to have continuous algorithmic outputs. So labs will need to integrate this data, record it, analyze it, and report it in a way that makes sense to regulators and that is always also compliant. This means higher reporting and compliance demands for labs. The second challenge is multiple methodologies. All these new methodologies, NAMs, are going to be coming with different testing methodologies. And this will create different workflows. Labs will need to validate new testing methodologies that come with their own documentation and compliance requirements. They'll need to create workflows to support these methodologies. And for the foreseeable futures, labs will also need to operate in a hybrid phase where they have animal models as well as NAMs. So the challenge here is very significant. And lastly, there's also all these testing methodologies will be introducing new instruments as well as specialized software. So this may mean that at the lab level, you can have, or you may have data that is trapped in one system and needs to be reconciled with a different system. If this data is not reconciled, you stand the risk of creating data silos and fragmented data sets. As you may know, manual reconciliation methods are often cumbersome and may even introduce more errors. For researchers, this may mean that the entire research is derailed or even sabotaged. And for the lab, this may come with financial implications, compliance issues, and even reputational damage. So what can the lab do to prevent these data interoperability challenges? And this brings us to the need of digitization. A few things are clear at this point. Manual processes are no longer sustainable as labs transition from an animal to a hybrid model and to eventually to a NAMS model. Automation ensures efficiency as well as accuracy, and digitization makes compliance and collaboration easier. So with this, why do modern labs need a laboratory information management system? And here I'd like to ask one question: how can a lab manager get ahead of all these changes that have been introduced by the FDA, Modernization Act 2.0? And there's one answer to this: acquire a robust LIMS. The FDA Modernization Act 2.0 drives complex data generation and compliance demands. It's clear that manual or outdated tools increase error risk, inefficiency, and may bring about compliance issues. So let's look at some of the ways in which a LIMS supports NAM workflows. And the first one is with a robust LIMS, your lab should be able to integrate heterogeneous data sets from the different NAMs that you have adapted. The LIMS will not only create seamless workflows, but it will also be able to embed SOPs, ensure end-to-end traceability, and also provide other quality assurance features to make sure that compliance, even with all these new instruments and software, compliance is maintained along the way. Third, a robust modern LIMS should be able to support instrument and software integration to ensure that you don't have any fragmented data sets or data silos. What do you do when you have teams working in different locations? That's where a cloud-based LIMS comes in. With this kind of LIMS, your lab will have real-time access from whatever location. And these will also ensure that the different teams can collaborate easily without there being any data silos or miscommunication. And last, with clinical research, security is very important. You don't want to have any data privacy breaches. This can be very costly. So a robust LIMS should be able to provide your lab with enhanced security features, such as multi-factor authentication and role-based access, among others. So at this point, we want to look at what features should you be looking for when you're choosing a LIMS to support your lab as your lab makes the transition or tries to implement the changes that have been introduced by the FDA, Modernization Act 2.4. And you're going to be looking at 7 key features to look for. And the first one, of course, you want a LIMS that offers robust data management capabilities. From the time samples are registered in your lab, the LIMS should be able to record all the data and metadata attached to these samples, analyze, record, and even eventually to the point where you're reporting the data. Other data enhancement features to look for include trend analysis reports, customizable reporting templates, as well as reports to flag out of spec results. These are all features that you should be able to find in a robust LIMS. Second, you want a LIMS that supports your workflows and ensures traceability. Third, you want a LIMS that offers enhanced security features such as secure audit trails, multi-factor authentication, role-based access, among others, including regular security updates. You also want a LIMS that can integrate all your instruments and software. Number 5, you want a LIMS that is customizable. With NAMs, from time to time, you might need to validate new testing methodologies, and you don't want to have to call in an expert to custom code, as this may come at an extra cost. And number 6, you want a LIMS that's easy to deploy and takes a short time, maybe a couple of weeks, as opposed to months. And you want a LIMS that's scalable. And here it's important to have a cloud-hosted LIMS that will also offer you remote access. And last but not least, you want a LIMS that is cost-effective. At this point, I'd like to mention that many people assume that building a LIMS from scratch is always the cost-effective option. However, it's important to have a big picture mindset. Do your math, see how much it's going to cost you to secure infrastructure, to put up the LIMS, to call in experts, perhaps to do the design, then also to have a team in-house to keep the LIMS operational. Look at all the updates and customization you will need to make from time to time. Also look at the features that these LIMS will offer you, and then compare to a commercial LIMS that is paid for perhaps on a subscription basis. And then do the math or a projection of a 3-year period or a 5-year period. With this figure, you will be able to make a prudent decision. And this brings us to the end of our discussion today. If you forget everything else, here are 3 things to remember. And the first one is that the FDA Modernization Act 2.0 removes the animal first barrier. While this does not outrightly ban animal testing, it takes away the monopoly animals have had in preclinical studies. It also paves the way for the use of non-animal models that are more human relevant and more predictive of human biology. Second, NAMs improve oncology research outcomes and ethics. And lastly, labs must adopt workflows and informatics in order to remain competitive, even as we move to the adoption of this new act. At this point, I'd like to say thank you for being a very attentive audience.